Welcome student. In the last lecture of our fourth week, we have discussed about different tissue systems. We have discussed about the conducting tissue, supporting tissue. Now we will try to discuss some special type of tissue which perform secretory functions. Also under secretory structures, we will try to identify what are the different tissues which will perform this function. Now, when we talk about secretory tissues, basically they are the glands. We say them as basically as glands also. They are the one which are secreting some compounds, and they are responsible for production and secretion of those special chemicals, and these special chemical will serve various functions also, and they will also provide extra benefit, and they will help in pollination, they will help in protection, they will help in defense. And these type of structures are widely distributed in vascular plant. Most of the vascular plant, most of the structures will have some sort of secretory structures, either a very elaborate type of secretory structures made up of multicellular cells, or even a single cell could also serve as a secretory role. Now, they are extremely diverse. Diverse means they are of different types. They could be located anywhere in the plant. They could be located on the outer layer. They will be located in the internal tissues. Then they will be composed of like one cell or numerous cells that we have discussed. Then they will release secretions that might be present intact in the cellular structure, or they might be released to the external environment. And the secretions could be fluid in nature, or they could be viscous in nature, and they could be composed of various type of secondary metabolites. Now, they could be present in isolated patches, or they could be found in well organized structures. See that I have told you that they could be even a single cell also, or they could be specialized into some special glands which can perform the function of secretion. Now, the different secretions which they are releasing could be resins, okay. These are sticky substances. They could be essential oils also, like many essential oils you would have heard of that they are extracted from plants. They are also synthesized and released by these secretory structures. Then mucilage could be there, then latex also we have studied, heterogeneous compound like milky latex is there, it is found. Okay, so many different types of substances could be present in these secretions. Now, these secretory tissues could be broadly classified into external secretory and internal. See, external secretory system means these will be located on, or they will be located on the epidermis, or within the epidermis, or between the epidermal cells. These secretory tissues could be there, or they will be, or just below the epidermis. Okay, they may be unicellular or multicellular. Now, in an internal secretory system, tissues, definitely they will be located in the inner tissues, like in the cortex regions or in the pith regions also sometimes. So, they will be present in the inner tissues. Now, coming to the external secretory tissues, okay. First important external secretory tissue is glandular trichomes, or they are also known as hairs. See, trichomes we have already discussed in our previous lectures that they are found on the surface, okay. On the epidermis you will find lot of hair-like structures, okay. Hair-like structures, they could be unicellular, they could be multicellular, or they could be branched also, okay, like unicellular trichomes could be there, okay, or multicellular or branch, and glandular trichomes are there which are secreting some chemicals. They could be found on the above ground parts of plants such as stem, leaves, bud, scales and reproductive organs. Generally, like if these some globular like structure will be present at the tip and a stalk will be there. So, they will form a glandular trichome. Coming to coleoteres, they are also multicellular glandular trichomes which are found on the leaf or bud scale and that generate a resinous hydrophobic secretion. Now, coleoteres, generally the chemical compounds which they secrete are terpene in nature. Okay, terpenes are basically hydrophobic compounds and they provide defense against various pathogens like fungi is there, even against insect attack. These secretions provide protection. They are sticky in nature. Okay, so when an insect will come and coleoteres have secreted their secretions, they can stick to them and they can harm them in that way. They also reduce transpiration and help the plant to survive the arid condition. So, these secretions can also be found on the surface layer and definitely they will reduce transpiration, okay, they will prevent transpiration and will help in preventing desiccation. Now, stinging hairs are also special type of secretory structures which produce toxic compounds. These toxic compounds are stored in vacuoles and they are also a special type of glandular trichome. You can have the structure, these trichomes, they can have a stalk, okay, and at the end they will have a small knob like structure which will contain the secretion. Very common stinging nettles are produced in these plants and dendrochite and urtica. See, these plants have stinging hairs which have these secretions, and when animal come nearby to these plant, they will stick to it, and because of these needle like extensions, they will prick into the animal body and they will release these substances which are toxic, and then some sort of irritation will occur, and in this way they will prevent from damage, they will definitely prevent from herbivory attack. And then other important external secretory systems are nectaries and hydrothodes. So, that we will discuss what are these. When we talk about nectaries, see, these are secretory structures which contain sugary substances. Okay, they contain sugar solution. You all would have heard of that nectars are produced from flowers and insect roam around those flowers because of those nectar, to get those nectars, and lot of many other things they make from those nectar. Okay, it's a reward, you can say that when insect is coming, it is helping in pollination, and because of that help it is being rewarded by giving them nectars. So, nectaries are generally found in associated with floral structures, because insects will definitely come towards the floral structure. Flower is a reproductive organ, so they will contain those reproductive structures. They want the pollen grains to be pollinated, and insects are one of the agents with which the pollen grains are pollinated. Like they have attractive petals to attract they have multicolored petals to attract the insect, and along with that they also secrete nectars to attract the insect. Now, these which are associated with flowers are known as floral nectaries. However, these nectaries could also be found on other parts also, like leaves and stems, so they are known as extra floral nectaries. They will be found on stamens. Suppose if it is found in stamen, then it is known as intrastaminal nectaries. Or they will be found at the base of the ovary or the top of the ovary or sides of the ovary. Even in the petals also they will be found. Now these figures just show you that how these nectaries are present. Now you can see a petal here, okay, ranunculus species. These are the petals of a flower of this ranunculus species, in which you can see that the nectaries are present in the form of nectar pocket. Okay. So every petal will have a nectar pocket. That nectar pocket will contain nectaries. Similarly, in this plant also you can see presence of this is actually Thivacea. Okay, it's a yellow color flower. You can see that at the base of ovary, see from here the ovary is present and it is surrounded by it is encircled by a nectar, okay, a ring of nectar you can say. This is also a nectary. So yellow color nectary, you can and it surrounds the ovary. Here you can see the ovaries coming out, style will be there and stigma will be there, okay. So all these things will come out from here. Similarly, this is also in Catharanthus roseus. You will see nectaries are present around the ovary. See, two nectaries are present on the sides of the ovary. So here also you will see the ovaries here, and along with that style and stigma will be formed here. So in many plants these nectaries are present attached with the floral organ. Now the next one important external secretory structure is the hydrothode. Now hydrothodes are pore from where the water droplets or the water is exuded out from a plant. Now here you can see the figure shows that the water droplets are coming. These are modified pores which are found on leaf. These hydrothodes are made up of group of living cells which is known as epithem. See, suppose this is a pore which is found at the tip of the leaf. Now it is surrounded by definitely some outer layers. Outer epidermis is there. Now this pore and these cells which surround the hydrothode are known as epithem, and these are made up of lot of intercellular spaces, and just below these epithem you will have xylem, because water is definitely will come out from the pass through this xylem, and then they will come out and with the help of these, following the pathway of these epithem, they will be released to this pores. And this process of these exudation of water droplets is known as guttation. These, you can say that they are somewhat modified stomata which are permanently open, and because of which water is coming out from these pores. Then you will have salt glands, basically in halophytes, that those plants which live in high salt condition have salt glands. Their root environment, the surrounding contain lot of salt. So definitely that salt is toxic. So they have to release those extra salt. So they will have salt glands, and these salts you can even see they are present on the leaf surface in the form of crystals, and these, if it is not removed, then it will be toxic and it will affect various physiological process of a plant, like in mangrove trees. Mangrove plants which live in marshy condition and lot of salt is also there. So these have salt gland. Then coming to the internal secretory tissue. Now internal secretory tissue is embedded inside the plant. Inside the plants, and you cannot observe them by visualizing from the outside, because they are present in the internal. You have to cut the sections and then you have to observe them. Here you can see a picture where some sort of duct is present, some maybe a resin duct or some other oil cavities are there, but this is a microscopic image showing some sort of duct. Now, these internal secretory structures could also be of various types, like oil cavities are present. So, from the name itself we can make out that these cavities are filled with oil, okay, and they are lined by epithelial cells. See, these cavities are there, okay, and they are lined by some cells which are known as epithelial cells. Now these epithelial cells will produce these oil and they will store up in these cavities. Now these oils are very important to provide protection against herbivory attacks. So they have anti-herbivory nature. Now you would have observed these, actually many of the oils have a lot of medicinal properties and they have lot of aromatic smells also. See, like they could be terpene in nature, basically hydrophobic compounds are there. So lot of various chemical composition can be there of these oils, and they have a lot of they have aromatic smell, and you would have smelled the leaf of some like lemon tree leaf or myrtaceae eucalyptus leaf, and you would have observed a special type of smell from these leaves. So it is because of the presence of oils in these leaves, and they are commonly found in Myrtaceae and Rutaceae family. And these oils which are present, many a times they are also known as essential oils. Okay, you would have heard this term essential oil. So this is also given to the oil secretions which are coming out from the plant, and they have lot of medicinal value. Lot of oils you would have heard of also, like eucalyptus oil is there, then rose oil is there, many clove oil is there. So many oils are there which have medicinal property. Now coming to another important internal secretory substance tissue is resin duct. Now resin duct is filled with secretion and they are also lined by epithelial cell. Now the resins, basically they are a sticky substance and the chemical composition is terpene in nature. Now these type of ducts could be found in cortex, in primary and secondary phloem, as well as in pith, also in secondary xylem of stems and roots. Many cases resins are very common feature of gymnosperm. However, in angiosperm also they are present. Now another special type of secretory structure is known as idioblast. Okay. Now these Idioblasts are specialized secretory cells, and these cells are found dispensed in the normal cells. See, basically you can say that these idioblasts are cells which are found distributed between normal cells, and these idioblast cells can have isodimetric structure, or they might be elongated, or they might be tube-like, and they generally store tannin, mucilage, oils as well as crystals. Like you would have studied, like in the previous lecture we have discussed about some idioblast which were containing lot of crystals. Now the crystals could be calcium oxalate crystal, okay. Many other crystals could also be there, and their shape can be variable. These crystals could be needle shape, or they could be of various, like they could be they could be star shape. So in various shapes also these crystals are found, and they are found inside the cell. Suppose this is an idioblast cell. So in that crystal, in that cells also you will have those crystal, and this is the idioblast. Graphite type of crystal has needle shape, okay. Drusa type of crystal has various different type of shapes, and they have pointed facets. Now lithocyst is also a type of idioblast. You can say that it's a type of and it is found in the epidermal cell. That also we have studied before, that some of the epidermal cells enlarges, and inside that epidermal cell you have lot of crystal deposited in the form of grape like fashion, and these crystal are known as cystolith, and that special type of epidermal cell is known as lithocyst. Now this was the discussion about the external and internal secretory structure. We can also divide them on the basis of their origin. We can divide those secretory structure as those which are having lysogenous cavity and as well as schizogenous cavity. So when we talk about lysogenous cavity, means how they have originated. So lysogenous means somewhat lysis, it is related with lysis. Now some of the cells which are filled with secretory content, they are lysed out, and they form a cavity like structure, okay. So you can see here that many of the cells which were containing lot of secretions, okay, they all broke and they all lysed and they form a single cavity. Now this type of cavity is found in the member of Rutaceae, like in citrus, okay, lemon, all those things are there in Rutaceae family. Now in schizogenous cavity, it is formed by the separation of cell by the breakdown of middle lamella. So that also we have discussed, that how the cells are connected together, and if you dissolve this middle lamella, okay, the cells, the common wall will actually get dissolved and they will form a single cavity like structure. So here also these all walls would have been dissolved, okay, and then these inner cavities formed. Okay, and this inner cavity is filled with a common, these are is lined by some cells which are known as epithelial cell, and it is common in Asteraceae family, coniferales, some gymnosperm and Apiaceae family, okay. Then coming to next type of secretory structure is laticifers. See, laticifers is filled with latex, okay. Latex is a very heterogeneous type of chemical compound. You would have seen milky latex, see that all we have discussed before. Milky latex or yellow colored latex is present. So like here in jackfruit you can see the secretion of milky latex. Now, you can have two types, non-articulate laticifers and articulate laticifers. They are basically known as lattice cells, and they originate from a single cell, okay, and they might be branched or unbranched, whereas articulate laticifers, they are known as compound because they originate from multiple cells. So, that is the difference, that one is coming out from single cell and the other one is coming out from multiple cells. So, they form a compound laticifers. Another way of distinguishing them is articulate anastomosing cell, that is lateral connections are established with similar laticifers to form reticulate, like many cells are there, okay, and they will have some sort of lateral connections between them, whereas in another one no lateral connections are established. So, that is all about secretory structures, and we have seen here that how these secretory structures contain lot of various chemical composition, and they also help in performing various function, like latex where they are all, like gums are there, resins are there, mucilage, they secrete mucilage. So, they all help in defense and they all help in various like pollination in some other process. So, they are also very important part of plant structure. So, now thank you for this, and with this we end this module, and in the next week we will start with another topic.